Hey, welcome back to my YouTube channel. Today we're going back to 1952 into a London lab to talk about a discovery that changed biology forever, and a woman who almost didn't get the credit for it. What if I told you the most famous discovery in biology wasn't a light bulb moment? It was built on someone else's data. For decades, textbooks have handed the discovery of DNA's structure to two men, Watson and Crick. But they were missing the one piece of evidence that made the whole thing possible. That piece was photograph 51, and it belonged to a woman named Rosalind Franklin. So, who was she? Franklin was a trained chemist and crystallographer, an expert at firing X rays at molecules and reading the shadows they leave behind to figure out their shape. In 1951, she was recruited to King's College London to sharpen exactly this technique on DNA fibers. And she was known for being meticulous, the kind of rigorous, patient lab work that can take months to produce just one usable image. Let's break down how this actually works, because it's the foundation for everything that follows. First, you fire X rays at a fiber of purified DNA. Those rays scatter off the molecule's atoms in a pattern shaped by the molecule itself. That scattered pattern hits photographic film, leaving behind a set of spots. And then, this is the hard part, a trained eye has to read that pattern and figure out the 3D structure hiding inside it. That last step is where Franklin had almost no equal. In May 1952, she captured an image that would later be called Photograph 51. It showed a clear X shaped pattern, and to someone who knows how to read it, that X is basically a signature. It's the fingerprint of a helix. A tangled or flat molecule would never produce that shape, only a twisted spiral one would. Franklin recognized this immediately, and from that same image, she was able to calculate the molecule's width and the exact distance between each turn of the helix. So here's what her data actually proved in three parts. 1. It's a helix. The X shaped pattern confirmed DNA is twisted into a spiral, not flat, not tangled. 2. The exact measurements. Her precise numbers confirmed DNA is a double helix, not the incorrect triple helix model some scientists had been guessing at. And 3. The backbone sits on the outside. Franklin's data showed the sugar phosphate backbone forms the outer rails of the molecule, with the base pairs tucked safely inside. That third point turned out to be one of the most critical clues in the entire discovery. Now, here's how that data actually traveled. In 1951, Franklin joins King's College London. By May 1952, she's captured photograph 51. Then, in January 1953, without Franklin's knowledge, Maurice Wilkins shows that photograph to James Watson. The next month, Franklin's own unpublished measurements reach Francis Crick through an internal research report. And by April 1953, Watson and Crick published their double helix model in Nature. Franklin's paper appears in the very same issue, but positioned to look like supporting evidence rather than the foundation it actually was. Here's the twist most textbooks leave out. In 1962, Watson, Crick, and Wilkins won the Nobel Prize for discovering DNA's structure. Rosalind Franklin's name wasn't on it. Part of that is a hard truth about how the Nobel Prize works. It's never awarded after someone has died, and Franklin passed away in 1958, 4 years before the prize, from ovarian cancer at just 37. But the deeper twist is that even while she was alive, her role was minimized, and some later accounts focused more on how she carried herself than on the science she actually did. So, how has history tried to correct this? Biographers, people like Brenda Maddox, spent years piecing together the full record of what Franklin actually contributed and reshaping how we remember her. And here's a genetics mystery to leave you with. Franklin independently calculated that the phosphate backbone had to sit on the outside of the DNA molecule, one of the single most critical insights in solving its structure. She was reportedly just weeks away from publishing her own complete model when Watson and Crick released theirs first. So here's the takeaway: Rosalind Franklin didn't stumble onto the double helix by accident. She earned it, image by image, calculation by calculation. The story of DNA's structure isn't just Watson and Crick's story, it's hers too. That's not a footnote, that's a foundation. If this changed how you think about one of the most famous discoveries in science, hit subscribe, because next week we're diving into another genetics mystery hiding in plain sight. Drop a comment below. Who's another scientist you think history got wrong? I read every single one. See you in the next video.